Albert, I would like to start with you, please, if I may. Could you please tell us what you're truly doing to control dengue from your own aspect with the World Mosquito Program?
Of course, Hasim, and thank you for having me for this discussion. I think it's really imperative that dengue is put on the forefront of our efforts against disease control. Now, the World Mosquito Program is a non-for-profit organization. We have started our operations around 2011, 2012. And what we've done is we've developed a truly innovative and disruptive technology called the Wolbachia method. And the Wolbachia method works by infecting Aedes aegypti mosquitoes with the Wolbachia bacterium. Wolbachia is a naturally occurring bacterium that is found in around 60% of all insect species, except Aedes aegypti. When you infect Aedes aegypti with Wolbachia, what we found was that these mosquitoes are no longer able to transmit diseases like dengue, Zika, and chikungunya. The other unique aspect of Wolbachia is that it's a self-driving solution. So Wolbachia-infected mosquitoes will perpetuate the infection throughout native populations. So the way in which we approach this is we identify at-risk communities, and we engage with these communities on a very intimate level, if you want to do that. So we ensure that communities are on board, are across with what the technology is, and then we proceed to release mosquitoes within these communities. We release Wolbachia-infected mosquitoes, and these mosquitoes then replace the native population. Because it's a self-sustaining solution, you only have to do this once. Wow. And once you've done this, you can extract from your release site, and the solution will stay prevalent in the communities. And these communities are then protected against dengue, Zika, chikungunya, yellow fever. So we've started this approach in our own backyard in Australia, in Queensland, in 2012. Queensland is now essentially dengue-free. Wow. So we have eliminated dengue in northern Queensland. We then started to build up, and we have now reached a global target of around 14 million people protected as of 2024. We are in 14 different locations globally, and we have estimatedly probably prevented around 750 to 800,000 cases of dengue in the time that we've been operational.
This is truly impressive. Well, Dr. Wolf, you're truly known around the world as a virus hunter, and you was recognized by several awards for your basically forecasting for pandemics by looking at viruses and particularly emerging and re-emerging viruses. But I would like to have your insight. How do you think these kind of technologies will be able to tackle vector-borne diseases, something like malaria, for example, particularly where they are endemic in the world?
Yes, and I think part of the way I think of it is to look at how we're doing surveillance now for other viruses and other diseases. And one of the approaches that's being used is wastewater surveillance, which permits us to understand what's going on, for example, in COVID by looking in sewage, since you find COVID in sewage. Now the question is, how would you be able to use some of these new molecular approaches or engineering approaches to address vector-borne diseases, which you're unlikely to be able to see in sewage surveillance? Now, one idea, it's a bit sort of more DARPA-like. It's something that you would have to do a lot of fundamental research on, but I think would be quite interesting, is you certainly could imagine engineering mosquitoes to express CRISPR-Cas13 and selectively in such a way that would identify when a mosquito was specifically infected with a particular virus or even a viral variant, and then potentially lead to a reporter fluorescent protein that could be produced. And then you could imagine identifying those, because there's different promoter proteins and different fluorescent promoters that you can have. So you could imagine being able to even have kind of broad automated surveillance that would look at things like the diversity of malaria or the amount of malaria present in a community that wouldn't require the kind of collection and ongoing PCR, which makes for sort of difficult work, I imagine, what you find.
Fantastic. Well, Albert, with the World Mosquito Program being rolled out in 14 countries, and you just said you're targeting tens of millions of people already, apparently there's a lot of challenges and it's not an easy kind of job, and there's a lot of lessons to be learned from your experience. Could you please tell us your experience in this regard? But excuse me, but before you do so, I would like to ask Control, please, to play video number 1 for the WMP.
Global health emergency. 3 billion people are at risk for mosquito-borne diseases.
The haunting memory of dengue is the body ache.
Empecé mareándome, vomitando sangre.
The fever was, I really cannot explain.
We can now protect communities from the suffering caused by dengue, Zika, chikungunya, and yellow fever by releasing mosquitoes that carry the safe and natural Wolbachia bacteria. Mosquitoes that used to spread diseases can now prevent them.
Sesudah ada itu dipasang ember untuk penetasan itu, warga di sini nggak pernah ada lagi yang kena DB.
We successfully implemented our groundbreaking technology with communities around the world, reaching more than 3 million people in 10 countries.
Far North Queensland is now essentially a dengue-free area for the first time in well over 100 years.
E que eu acho que deve ser replicado em vários outros lugares, aqui no Brasil, na África, em qualquer lugar do mundo, dá certo.
When we release it that day, it like we are releasing hope.
Now we're ready to take our groundbreaking solution to the next step, to protect 100 million people around the world, together.
Please.
Yeah. So I think one of the important messages, both from this video as well as some of the earlier comments, is the scale of the problem. 4 billion people are at risk of dengue and contracting dengue. And to be able to protect 4 billion people is a daunting task. And it's not a task that the World Mosquito Program can do by itself. It's not a task that I think any company or any organization can do by itself. So one of the key lessons here is that we need partnerships. We need to engage with governments. We need the technology to be embedded in private, in government's health initiatives to be able to reach as many people as we can. So mosquito production is difficult. Producing that many mosquitoes is not an easy job. A mosquito is a fairly sensitive little creature. As annoying as they are, they're very hard to produce at scale. To develop the infrastructure and the expertise that's required to roll out this solution requires funding, requires commitment, and it requires buy-in again from governments. So key message here is we need government buy-in to be able to do this. The other key message is around communities and community perception. The technology is very innovative and it's disruptive. We are releasing biting insects in people's backyards. So you need to be able to communicate very clearly and effectively on the benefits of the technology, why we are doing this, and you need to engage and make sure that your community is on board and that the community is driving this innovation and the technology. You cannot be a foreign company or a company from, from somewhere else trying to do this over the community. You need to be part of the community. And for WMP, what we've learned over the last 10 or so years is that the success of this technology really is dependent on the community being part of the solution.
Fantastic. Thank you so much. Well, I would like, you know the story, guys, but I would like to tell the audience about a small secret, actually. Back in 2012, I've attended a talk by Scott O'Neill, who is the CEO of World Mosquito Program. demonstrating the impact of tackling dengue through Wolbachia. It was truly innovative. And then I visited his insectary at the University of Queensland. It was quite something from the future, where you see cages of mosquitoes flying around. And that's something was extremely innovative, particularly when you see how such a public health threat is being tackled. And of course, you know, to me that was like a sci-fi. But I truly got goosebumps when I knew that this technology is actually exists in Saudi, implemented. And that is just demonstrating how our vision is truly being translated into such an impactful work. And the person who's leading Saudi Arabia's effort is Dr. Assam Tawfiq. And I would like to understand from you, Dr. Assam, how did you attract this technology to be implemented in Saudi from a strategic point of view? This is a deserted area, part of the world. We don't get a lot of flood. It's not like Brazil. But why?
Well, King Abdulaziz City for Science and Technology has always been driven by technologies. It also addresses the national challenges through technology-based solutions. After the MERS-CoV era, dengue started to raise as the next potential healthcare challenge here in the kingdom. So our technical team back in 2016 started to scout for emerging technologies that can actually be utilized to control this disease. After a thorough analysis, our technical team identified Wolbachia as top of their list.
Wow.
So we contacted our counterpart in Australia, which is CSIRO, who they introduced us to Ari Hoffman from University of Melbourne. So Ari and his team first visited us in 2017, and in early 2019, we signed our partnership agreement with them, and this is how it all started.
Fantastic. Congratulations again for hosting this very impactful work. Nathan, from your book, The Viral Storm, you talk about the future of pandemics and how you, so I would like to understand from you, how do you think these kind of innovative solutions, such as, of course, the utilization of Wolbachia, but not necessarily it, also the utilization of genetically modified mosquitoes, for example, will be able to tackle vector-borne diseases outbreaks? And how do you think this will fit into the broader picture of infection prevention and control, as well as pandemic preparedness?
Well, I really appreciated the question. As a doctoral student, one of the things I was interested in was documenting the diversity of viruses in wild animal species, since wild animals are often the source of novel pandemics like COVID or HIV. And that can be a particularly challenging task. In some situations, you have to anesthetize and dart wild animals, or you can try to get remote specimens from feces, for example, or urine. But these are very, very difficult approaches. And one of the things that we attempted to do was to actually use mosquitoes that had fed on animals to try to get molecular evidence using PCR of viruses that or other parasites that infected them. And that was something that was quite difficult to get, because you would have to identify the animal and actually wait for the virus, wait for the mosquito that had recently fed on the animal to be able to collect it. But you could imagine the same sorts of tools we were talking about earlier, being able to potentially even have mosquitoes as a reporting system that aided in surveillance of wildlife disease diversity and viral diversity, using some of the same approaches we spoke about earlier. And I think obviously these are some ways off, but as you think about the kind of work releasing, you were talking about millions of mosquitoes. You really get to a situation where one could imagine blanketing areas to understand the full diversity of potentially pandemic viruses. I think that would be certainly an interesting use of the technology.
Wow. So it's obviously, from what I have understood so far, it's quite a big operation, really. It needs quite a setup. It needs, from all aspects, from infrastructure point of view, as well as, of course, from funding point of view, logistics, laboratory, trained personnel. In a nutshell, could you please, Dr. Isam, explain to us and educate me how this process is being done in order to roll out Wolbachia? But excuse me, before you do so, I would like to ask Control to kindly play video number 2 from Caxt to help you visualizing this process. تفضل.
Well, to do so, Dr. Hussam, we had to establish our biocontrol facility in Jeddah, where the problem exists here in Saudi Arabia. So this facility consists of a mosquito rearing unit. This is Abdullah Shamrani, who actually managing the mosquito colony room. And what he's doing is actually collecting the sandpaper that the mosquito eggs laid into after we inject them with the Wolbachia, of course. He's photographing these sandpapers for the purpose of counting. And after this, another technician will come and check the quality of these eggs under the microscope. This facility also capable of producing 100,000 mosquito per week. This is the capacity. But we are aiming to reach to 1 million mosquito when we, in the next phase, when we're going to cover as many districts in Jeddah. Another technician later on will actually transferring the mosquito pupa, as you can see, and place them into these cups that are stuck with QR codes. I will speak about the QR codes later. We feed them with sugar solution. I don't want to go technical here, but I'll try to make it as brief. We feed them with sugar to give them energy needed for reproduction. And before we release them, the mature mosquitoes, we feed them with blood. So they are ready for the release now. In the facility, we also have a pesticide unit. Actually, we utilize this pesticide unit to evaluate the mosquitoes in Jeddah environment. And unfortunately, we found out that the mosquitoes there resist 3 types of pesticides. So you can imagine the hurdle of keep using pesticides. We also have a molecular biology lab. This is Lama Al-Judaybi, who's actually managing this lab. And what she's doing, actually detecting the Wolbachia of the mosquito that we are collecting from the district after we release them in certain time points. She's using RT-PCR and LAMP technology for doing this. I don't want to also to acknowledge our national partners, Jeddah Municipality and Wiqaya, who provide us with full support during the project. In terms of logistics, Dr. Hussam, we have a dedicated team for the release and for the collecting the mosquito or the monitoring phase. This team led by Yasser will show up in a few minutes. So they release the mosquitoes in certain places in a district that we target, and then they scan the QR codes, and this will be translated to a platform that we created for this project to know exactly where is the location of the mosquito that we release. And yeah, and this is how we do it. And this is the mosquito after releasing it.
Fantastic. It's really great to see that Internet of Things and geospatial kind of work is being utilized to help monitoring the spread of these mosquitoes and make sure that your work is being done. But I'm sure the pressing question I do have, and I'm sure that many of the audience will potentially share that with me, Is this like a scientific experiment we conduct for basically intellectual curiosity, or is it truly impactful and that can have a dramatic change on public health level? Does it have any cost effectiveness? And I think the best person to answer this question will be definitely you, Albert, after the many years you have done and the many rollout you have conducted with using Wolbachia in many different countries. Please.
Thanks, Osam. I think this is the favorite part for me of the discussion because I get to share basically an incredible success story in terms of effectiveness. So over the years, the World Mosquito Program has built up a portfolio of evidence, of efficacy evidence, that demonstrates both the health impact as well as the cost effectiveness of the innovation and the intervention. So as the video mentioned, we've started in Queensland, in Northern Australia, and in Queensland, from public health data, we can demonstrate that we've essentially eliminated dengue. We didn't stop there. We've conducted, to follow up our portfolio of evidence, we've conducted a randomized controlled trial in Yogyakarta, in Indonesia, that ran from 2017 to 2020. This trial consisted of 2 arms. One was a treated arm and one was an untreated arm, test negative control methodology. And this RCT demonstrated that we were able to reduce dengue incidence in the treated areas by 77%. And we were able to decrease or reduce dengue hospitalizations in the treated area by 86%. So an incredible efficacy results. We didn't stop there. We continue in every release area where we are releasing mosquitoes to monitor public health data. And from that public health data, we've been able to build up the stratified portfolio of evidence. And our efficacy range in every release site that we've done so far, whether that's a partial release or a full release, range from between 60% to 98% reduction in dengue cases. Keep in mind that 2024 has been a record year for dengue. Like, this is the most dengue cases on record ever. And we've been, even in these like extreme dengue occurrence, we've been able to demonstrate almost 0 dengue cases in our release areas. So a really effective, cost-effective intervention. The fact that we only have to do the release once and we don't have to come back really has cost implications. We've done several economic studies that demonstrates, depending on what the socioeconomic situation of the release area is, but the intervention pays for itself within 5 to 10 years. And from that, you get a return on your investment as a return in terms of morbidity and mortality data. So
fantastic. It's truly impressive. Well, as biologists, I'm sure we all would agree that living creatures, do mutate in order to adapt to any external changes. And they know, Dr. Wolf, you're using single-cell genomics to understand basically the genomics of many single cells. So how do you reckon single-cell sequencing would actually help to understand the emerging pathogens, especially those when it comes to detecting rare species and strains?
Yeah, it's quite remarkable what's happened over the last 5 to 10 years from going from a situation where you would take a specimen and effectively all the cells would be, mixed together and you would sequence them and not be able to know exactly what had come from which cell, to a situation where we now can sequence from single cells. And I advise an interesting company called BioScribe that has the capacity, their technology permits you not just to sequence the RNA from individual cells, but RNA and DNA and potentially DNA methylation and other things from cells. These approaches, both within cells and also in larger specimens, have the capacity to have much higher levels of sensitivity and to get larger fragment sizes. So part of the problems we have now, as I was mentioning, the the wastewater surveillance is you often get you have low sensitivity, so unless there's a high density of the virus in the specimen, you often will miss it. And second of all, you'll get very, very small genomic fragments when you sequence those specimens. And so I think this next generation of tools is going to permit us to do much higher sensitivity surveillance, get larger fragment sizes, and eventually to be able to sequence individual bacteria and do some of the things that we've talked about in terms of looking at antimicrobial resistance, which of course is your area of expertise, and being able to, the idea of being able to get a full genome from an individual bacteria, I think is, we're starting to, the bioscribe technology is being used for soil specimens and starting to be able to do that. And I think it's just a matter of time before we're able to do it with human and other mammal specimens.
Fantastic. Well, during the closing remarks of the G20 summit, which Saudi has hosted, Sumu Sayyidi, His Royal Highness the Crown Prince Mohammed bin Salman, has demonstrated the country's ambition and commitment to combat the future pandemics, and of course put the country on the forefront to prepare for future pandemics. So I'm sure, please don't get me wrong, Dr. Assam, I'm sure dengue is definitely of clinical importance. But of course, with the great infrastructure you demonstrated with us, I'm sure there are many, many other vector-borne diseases that you would like to tackle. And of course, with the great support you will be having from different industries and organizations, and of course, including CACS itself. We are so happy to see the Vice President of CACS with us, Dr. Batul El-Baz, who's demonstrating, this is basically a testimony for the support all of the scientists at CACS and also in many other institutes would receive to combat many diseases, including communicable diseases, and of course, in this particular aspect, vector-borne diseases. So tell us, how would you expand and tackle other vector-borne diseases?
Well, Dr. Hussam, the ultimate goal of what we're doing now is actually to see the reduction of the cases of dengue fever in Jeddah using this technology. Hopefully, in the next phase, we achieve this ultimate goal. After that, since Aedes aegypti also transmitted different diseases, other diseases like chikungunya and yellow fever, which also exist in the kingdom, but at very low prevalence, we also want to explore the effectiveness of Wolbachia against such diseases. CACS is also committed in building capabilities. As speaking now, we have a top scientist, a top researcher, who's doing his PhD in Imperial College of London about gene drive. So but not against infectious disease per se, but this will be another tool that we're going to utilize when he's back, inshallah, in a year or so, against vector-borne diseases. At the end, we want to achieve what is the mission of RDIA, of course, in reduction of infectious diseases in 2035 by 50%. And hopefully, as a national endeavor, we all can do this and achieve it.
Fantastic. So definitely, I'm sure now you would agree with me. As I promised you earlier in my opening speech, there's hope. And of course, this hope is being demonstrated by the leaders in this endeavor. So I'm truly energized and optimistic that with this utilization of bioengineering applications, we're going to shape the future of the global health. So please join me to thank our distinguished panelists and give them a round of applause for not only for participating in this session, but also for the impactful work they are leading. Thank you so much.
Thank you.